Good evening. My name is Scout Meredith Best, and I am a student project manager for the Clark Forum for Contemporary Issues at Dickinson College. I would like to welcome you all to tonight's event, The Science of COVID-19, Aspects of Infection, Immunity, Treatment, and Testing. With the majority of American citizens living in shelter-in-place orders and over 1 million cases of COVID-19 in the United States alone, we have all seen the news that addresses the impacts and the victims of the pandemic. But with such a plethora of facts, figures, guidance, and stories circulating at rapid speed online, it can be difficult to comprehend the scientific aspects behind COVID-19. Tonight's event will address our current understanding of how the virus harms our bodies and why it has been called the perfect pathogen. So without further ado, I would like to introduce tonight's presenter. John Henson serves as both the Senior Associate Provost for Academic Affairs at Dickinson College, as well as the Charles A. Dana Professor of Biology. He is a broadly trained cell biologist and comparative immunologist. His work and course offerings at Dickinson have focused on infection versus immunity, biochemistry, and molecular biology. On a broader scale, he has worked on the H5N1 pandemic influenza preparedness, along with biological weapon nonproliferation, while William C. Foster Science Fellow at the U.S. State Department. Before beginning, I would like to take a moment to thank those who are on the front lines of the pandemic. Your work and sacrifices to ensure the well-being of so many are deeply appreciated. I would also like to thank our viewers for being here tonight. These are trying times for us all, and I hope you are all staying safe and following recommended safety guidelines. There will be a question and answer session immediately following the program, so please type your questions into the live chat next to this YouTube video at any time. Sarah Duane, Clark Forum Student Co-Supervisor, will be moderating the questions. We will now begin the presentation. Thank you.
Hello, and thanks for joining us. As you can see, I'm speaking from home tonight, like I'm sure you all are sheltering in place. And I'm going to try to sort of summarize at least my understanding of the science, in terms of kinds of things that I've been able to read and look at and try to comprehend. And we're going to start at a very basic level, and a lot of this talk will be done with slides, so I'm going to go ahead and share my screen and start that process. All right, so this is actually a scanning electron micrograph of a cell that is infected with SARS-CoV-2 is the actual virus that causes COVID-19, and these are the little red things that you see here is actually the virus itself. And if we start very basically, the sort of central dogma of biology is that we have this inflammation flow where we have, of course, the information molecule of DNA, which of course serves as a template for RNA, which then serves as a template for protein. And we think of that information flow as a one-way process, 3 steps in our cells. But it turns out that many viruses can do this process. So for example, the chickenpox virus that you guys may have had when you were younger, that virus actually can infect cells and go from a DNA level to an RNA level to a protein. But a virus like HIV, which is an RNA virus, actually can replicate in RNA, but also can revert itself to DNA and then insert itself into the DNA of the host cell. It turns out that SARS-CoV-2, the virus that causes COVID-19, that's a virus that is only an RNA virus. And so it's an RNA virus that replicates in RNA and then also makes protein from the RNA. All right, so now it's strange. Okay, I'm just trying to get my mouse back. Here we go. All right, so the SARS-CoV-2 virus is seen here in clear my drawings, in an electron micrograph. And you can see, as all coronaviruses, it has these very prominent spikes on its surface, which constitute the spike protein. The spike protein is then diagrammed in its molecular form here in red, coming off the surface of the virus. And like all viruses, it needs a receptor to interact with the surface of the cell in order to gain entry. Remember that viruses can't really replicate on their own. They need the machinery of the cell to do so. And as an RNA virus, once it gets inside the cell, it actually then will make proteins from its RNA as well as replicate its RNA in order to reconstruct a new virus. So SARS-CoV-2, the actual receptor on the surface of the cell that it binds to is a protein called ACE2, or angiotensin converting enzyme 2, which is a protein that's actually involved in blood pressure regulation in the body. And it's a protein that's expressed by quite a few cells. It's expressed, has a lot of cells in the respiratory tract and the digestive tract, in the blood vessels, in the heart, and the kidney. So there's a lot of places where you find this ACE2. As it happens, there's another protein that's involved in the entry of the virus into the cell, and it's called TMPRSS SS2. It's actually a protease, or a protein that actually breaks down other proteins. And it turns out that the virus protein has to be broken down a little bit before it's able to actually enter into the cell and escape from the cellular membrane that it's encased in to actually release its virus, makes more of itself and its viral proteins as it goes along. So where does the SARS-CoV-2 virus first infect the cell? Well, there was a paper that just came out a couple of days ago looking at what cells express these proteins that are known to be receptors and necessary for SARS-CoV-2. And it was noticed that the nasal epithelium, in other words, the layers of cells in your nasal cavity that separate you from the air passages, actually are relatively rich in ACE2 as well as this TMPRSS2 protein. And so those are likely cells where the virus is going to land and then it's going to take up some residence. It also might explain the notion that we know that folks who have COVID-19 and shedding viruses, they tend to be relatively infectious. So that would make some sense that if the virus is in residence in your nasal epithelium, it's much more likely to then be spread to the outside world. Other coronaviruses, like this MERS virus that you may have heard of, the Middle East respiratory virus, actually it impacts cells with receptors that are much deeper in the respiratory tract. So it's less likely to be transmissible than SARS-CoV-2. So let's follow the respiratory tract down into the place where things have to take place in order for you to breathe. We know that as you follow air passages in the lungs over here, that they branch continuously into smaller and smaller branches. And at the very end, you can sort of think of them as a bunch of grapes, right? At the end of the actual branch point is a swelling, and that swelling is an air sac called an alveolus, and that's where, of course, gas exchange takes place. And those air sacs are actually covered with a series of capillaries where blood cells are running, of course, because what you're trying to do in your lungs, obviously, is exchange CO2 in your red blood cells for oxygen. And in order to do that, you need a very fine membrane in which that gas exchange will take place. So it's a very delicate organ in many respects. And these fine air sacs create this ideal intimate association between the blood and the air that allows for exchange of gases to take place. You can see some of these alveoli on the surface of this lung right here. And if we could look with a microscope at what you're seeing an alveolus looks like, this is a diagram of it. And so this is airspace here and airspace out here. And then in the wall of the actual air sac is the capillary with the red blood cells, as you can see them. And you notice that the capillary wall here, or the cell that creates the wall of the capillary, is not separated by very much from the wall of the cell that is making the airspace. And you can see that also in these light micrographs of the walls of alveoli in the lungs. They can be very, very thin, as you can see. And this makes sense because, of course, you're trying to afford the least amount of space for gas to exchange in this environment. And in an electron microscope, you can see this in much more detail and higher magnification. And in fact, here's a capillary. You can see the walls in this greenish gray. Here's a red blood cell. Here's another capillary. Here's a cell of the actual wall of the air, the actual cell that makes the wall of the airspace. And here is a very high magnification micrograph showing the airspace up here, the wall of the airspace pneumocyte, the cell that makes the space of the lung. Here's the cell that makes the wall of the capillary, and here is actually the red blood cell. So this is a tiny space from which gas can be exchanged. And this is the whole physical reason that you can actually breathe well, is because you have all these air sacs that allows for gas exchange to be very rapid, very efficient. And if something interferes with that, of course, that interferes with the ability to oxygenate your blood. And this is a series of micrographs that shows what's happening in patients who have things like COVID-19. So up here in the top row are a series of micrographs of healthy lung tissue. And as we've said, mostly what you see is airspace, and you see these very thin walls of the air sacs, which contain the capillaries as well as the pneumocytes. And you can see that across all these healthy lung tissue. Down here is a row of micrographs of lungs that have what's called ARDS, which is acute respiratory distress syndrome, which is a general term given for folks who are having inflammation and infiltration in their lungs, very similar to folks who have COVID-19. And as you can see, you don't have to be a pulmonologist to convince yourself that, wow, if the wall of the alveolar sac, which is supposed to be very thin to facilitate gas exchange, all of a sudden becomes incredibly thick and full of fluid and disruptive in terms of the actual space that these gases can go across, you might have trouble actually exchanging your gas and getting oxygenated blood. And sometimes the infiltrate of cells and fluid in the lung space can get very, very dramatic, in which the alveoli essentially go away because they're filled up with fluid. And at that point, gas exchange doesn't take place at all. So this is a very critical situation, obviously life-threatening situation, ARDS. And this is something that happens routinely in COVID-19 patients. And just to get that at a gross anatomical level, this is actually what are axial sections. So it's like you took a buzzsaw through essentially the middle of your chest where your heart is. So these are CAT scans through the healthy lungs here at the top. And so this is the heart right here. And just like with the micrographs, we expect to see a lot of air. Well, in this CAT scan, you would expect to see a lot of blank space because there should be a lot of air in a healthy lung. And there is, as you can see, it's all black here. So it's all essentially air. You see some airways and blood vessels and the like, but largely these are two different sections, axial sections. You can largely see as blank space. However, in a COVID-19 lung, as you can see down here, in a COVID-19 lung, what's happening is you get these infiltrates that appear in this sort of fine web work, particularly at the outer periphery of the lobes of the lung. This is often referred to as ground glass opacities by radiologists. So it's indicative of this filling of fluid and infiltrated cells in those actual areas of the lung. And of course, those areas of the lung are going to be much less gas exchange is going to be taking place because they don't have the actual anatomy to do so. And over time, this can get worse and worse and can progress to the point where it's life-threatening. Of course, as this happens, there's efforts to increase oxygen levels that are given to patients. Eventually, patients are put on assisted ventilation. And all that is tried to try to help with gas exchange. But it turns out that really, once you lose the ability to breathe, there's very few options One of them is extracorporeal membrane oxygenation, which is something that can be done to your blood, but that's very rare and is something that is much, much rarer than a ventilator. Let's put it that way. This is actually a healthy lung over here, seen more in a longitudinal profile. What you're seeing here, all the air passageways that you can see in the lungs here. And this is this COVID-19 patient showing again where those opacities are, these places in the lungs where there was fluid filled up and where this doesn't allow for gas exchange to take place. All right, so one of the questions is, well, what can we do in order to treat these kinds of viruses? And I just want to go back quickly to the original diagram we showed of the viral life cycle. And one thing we could do, of course, is block the viral receptors. And there are some drugs that actually interfere with ACE2. In fact, some of them involved in blood pressure regulation. There are some protease inhibitors that could be used for this Tempers RS2 protein. There are, of course, a whole suite of drugs that are involved in interfering with the replication of viruses. And remdesivir, which you've probably heard of, is this drug from Gilead Sciences that is actually one of those kinds of drugs. But there's a whole bunch of them that are involved in that kind of thing. There's also involved in viral processing of proteins, other protease inhibitors and the like. So there's a whole suite of antiviral drugs that are being looked at in terms of their efficacy in terms of treating COVID-19. And in fact, there's hundreds of these drugs being tried and hundreds of clinical trials going on right now. Well, what about your immune response to this? Because I know you've probably heard that sometimes that is also a problem as well as the good thing going on, of course. And it obviously is impossible to talk in any depth about sort of the whole immune response because it's something that's very complicated and involves a lot of different steps. But this one diagram is an effort to try to summarize what's going on in immunity in a very simplified way. You guys have all heard about antibodies and antibody-based testing and antibodies being important proteins that are produced by the immune system. As you can see, antibodies are these Y-shaped proteins, and they're actually produced by a lymphocyte type called B cells over here, and they're part of what's called the adaptive immune response, which is relatively slow. And adaptive meaning that it has the ability to adapt. In fact, the virus, the pathogen, really teaches the immune system how to respond. It adapts, it's highly specific. And another very critical feature is that adaptive immunity has memory. In other words, it can remember the pathogen into the future. You notice that there's another prominent cell over on the adaptive side here called a T cell. And the T cells are actually cells that serve as regulators of the immune response, particularly these so-called T helper cells here, these CD4 cells. There are also cells called T killer or cytotoxic cells that go around and kill viral and bacterial infected cells. So they're really important antiviral responses too. And in fact, T helper cells help B cells make antibodies. So T cells are incredibly important to the immune response. You don't have to look any further than HIV about their importance because HIV is a virus that homes in on T cells, particularly T helper cells, and kills them. And in that case, causes immunosuppression in people. But the adaptive response is relatively slow, right? It takes several days to develop. So in the meantime, the virus is replicating. What's dealing with the virus? Well, of course, there's a whole other group of cells over here that are involved in so-called innate immunity. In other words, they are less specific, but they tend to act relatively quickly. And notice that one of these cells, a classic cell involved in eating other things, is called a macrophage, and it goes around eating viral infected cells, eating a new virus, that kind of thing. Dendritic cells also are highly phagocytic, like to eat things. They're involved in actually taking up some of the virus and then, interesting enough, presenting it to T cells to help them sort of recognize it. That's part of the virus sort of teaching the immune response. And then we have these interestingly named natural killer cells. And natural killer cells, as the name would imply, are cells that go around and kill other cells. And they do that in response to viral infections. They do that without the specificity or lag that a T cell would. And so they're very important in the early stage responses to viral infections. And we have a bunch of granulocytes in the blood, things like neutrophils that are phagocytic, mast cells that are involved in actual release of pro-inflammatory mediators and complement, all that kind of thing. The point being that this side of the immune response is relatively quick and can work, but it's not necessarily as specific or adaptive or has memory like this adaptive immunity does. So this is a graph that is going to attempt to show what's happening over time as we look at days post-infection, and I'm just going to draw a few things here, try to at least. I noticed that I lose, oh dear. I don't think this is going to work, actually. This is not going to work. Let's try this. I'm going to escape from here. All right, so I'm going to go back to this and clear my drawing and then try to start drawing again. Here we go. Okay. So let's say immune response is just starting with a pathogen. And what I'm drawing here is the response of some early cells to viral infection. And these early cells, what they're actually releasing are a series of chemicals that are often interfering with viruses' ability to infect cells, and they're called interferons, interestingly enough, and I'll draw that in a very clumsy way. There they are, interferons. So they actually come up very quickly. They actually interfere with things like the ability of the virus to replicate. They also interfere with the virus to do protein synthesis, and so they're very important. And then at the same time, very quickly, what's happening is we have an increase in our natural killer cells, which are killing things off. Remember, they're killing things relatively non-specifically, right? There's our natural killer cells doing their thing. And meantime, of course, T cells and B cells are starting to recognize the viral targets, but it's taking them time to develop their response, because one of the things they do is they actually develop more of themselves. They divide to make more of themselves. And in fact, you may wonder, well, where does the specificity of the actual antibody come from? In other words, how does a B cell actually recognize these viruses? Well, believe it or not, you already have those B cells in your body now that recognize SARS-CoV-2, even though you've never been exposed to it. And the reason for that is that as you were developing as a fetus, your B cells were actually going through a developmental process in which they were going through random reassortment of their DNA to generate all kinds of specificities for antibodies. So in your body now, you have tens of millions of B cells, which have tens of millions of different specificities for their antibodies. And there's a good chance that some of them have specificity for SARS-CoV-2. And that's how it works. So once SARS-CoV-2 comes into your body, those B cells actually recognize it. They make more of themselves, and then they start secreting antibodies. So those antibodies over time are going to be secreted. And of course, they come up slowly at first. And you may have heard that some of them initially are called IgM, which are antibodies that are a little bit less specific. And then over time, what happens, so draw this, is you get antibodies that are called IgG. We'll make that a final color. What can we do? Brown sounds good. So that's taking even more time. And so here's IgG. Okay, so as you can see, the antibodies, which are sort of indicative of the adaptive response, are happening slower, right? Whereas the natural killer responses, the interferon responses are very quick. And if we were going to draw in a peak that showed specific T cell responses, it would go up largely in concert with these antibody peaks as well. So then what would happen if you re-exposed a person to the same virus? Well, what would happen, of course, is that you would have memory B cells and memory T cells that were left behind after the initial challenge. And they would actually then start without much lag period to make more of themselves and to make antibodies and to make T killer cells. So the notion that someone exposed to SARS-CoV-2 would not have any immunity to that. I think most immunologists would disagree with that notion, that there would be some level of immunity afforded to it. And that's just because of immunological memory, which is something that happens. Now, it's possible that folks have different levels of antibodies that they produce, different levels of T cell responses. But by and large, it would be expected that folks with antibodies against SARS-CoV-2 would have immunity. Why do I say that? Well, a number of papers have been published where they've taken convalescent serum, so serum from patients who've recovered from COVID-19, and they've tested their antibodies in vitro, in other words, in cell culture systems in which they try to expose cell cultures to SARS-CoV-2, and they show that those antibodies are neutralized. In other words, they have the ability to block the virus from binding to the receptor and not infecting the cells. And you say, well, that's in cell culture petri dishes. That's not very realistic. Well, they've also done the experiments with non-human primates. So they've done experiments with rhesus macaques in which they've taken convalescent serum from patients who've survived COVID-19. And they've shown that they actually can be protective in terms of neutralizing a challenge with SARS-CoV-2 in a rhesus macaque model system. So short of actually exposing patients who've recovered from COVID-19 with SARS-CoV-2 directly, that's about as much evidence as we can get without doing that. There's no question that it's possible that a person who's recovered from COVID-19 could get reinfected, but the likelihood of their infection being more mild is much higher probability. So we'll leave all that, I think. All right, so great. I'm gonna go to vaccines. Vaccines are something that everybody is talking about. And of course, vaccines, one of the things you think about them doing is creating antibodies, right? And so the vaccine essentially has to imitate the original virus. I think there was probably as much excitement about this vaccine for COVID-19 as there was in the 1950s and 60s for the vaccine of polio. And that's something that actually had galvanized the United States in terms of trying to actually generate a vaccine against polio, which was a devastating disease of children largely. And there were many different approaches, but two scientists that actually were competing with one another. One was Jonas Salk, who actually worked at University of Pittsburgh originally. And then the other was Albert Sabin, who was really someone who also was out of Cincinnati and was competing with Salk to try to come up with an appropriate vaccine. And the reason I'm talking about the polio vaccine is because it turns out that these two scientists were using two different approaches. And it turns out that many different labs are using different approaches today for vaccines against COVID-19. So Jonas Salk's lab was using killed virus. In other words, they would take the polio virus, they would expose it to chemicals, in this case formaldehyde, kill it, and then they would use that virus to actually inoculate children. And hopefully that virus would then, of course, elicit an immune response and antibodies to protect the child with subsequent challenge with the actual virus. Sabin was not convinced that that would work. He was much more convinced that if you had a weakened polio virus, a so-called attenuated virus, that if you exposed individuals to that, that would more likely generate a robust immune response that would be more protective. And so, as it turns out, Salk won the race, if you will, in the United States, and that killed polio virus was actually established in the US in the 1950s. And then Sabin, because he wanted to test it elsewhere, he went to the Soviet Union and actually did live virus polio vaccines there. And eventually Sabin's live virus vaccine replaced Salk's vaccine, although we come back to Salk again. The point being that vaccines take different strategies. There's pluses and minuses to different approaches. And the same thing is true right now with the vaccine developments for COVID-19. So for example, there are vaccines that are taking advantage of portions of the virus. So the spike proteins that we mentioned seem to be really important in terms of binding to receptors on cells. So there's actually spike proteins themselves that are being used as just a subunit vaccine. There's actually a recombinant spike protein vaccine coming out of UPMC that has now been tried in animal trials and is going to go into clinical trials. There's a vaccine that's come out of Oxford University, the Jenner Institute. Edward Jenner, of course, was the individual who invented vaccination, if you will, using cowpox to vaccinate people against smallpox. And of course, the vaccine itself is derived from the Latin vacca for cow, right? So the Oxford University group has taken the approach of using another unrelated virus, in this case a chimpanzee adenovirus, engineer it to express the SARS-CoV-2 spike protein, and then use that as a vaccine. Now, it turns out that they've already started relatively large-scale clinical trials. So for example, this week, they're actually doing a trial with 1,000 patients because they've already gotten good safety information on that particular vaccine because they've used a similar approach to vaccines they've tried on SARS, the 2003 SARS coronavirus. And they anticipate to have trials for efficacy, believe it or not, this is a phase 2 trial, to happen this summer, as soon as next month. And they anticipate trying to vaccinate at least 5,000 individuals in areas where there's active COVID-19 community spread to test for efficacy, which is an extremely rapid time frame, if you can imagine that. There are also other groups that are using other viruses as carriers, if you will, for proteins from SARS-CoV-2. And then there's groups that are using new vaccine approaches, for example, using messenger RNA that codes for the spike protein that's encapsulated in a lipid nanoparticle that is actually injected. That's actually the trial that was started by the National Institute of Allergy and Infectious Diseases. There's also DNA-based vaccines as well. And you may wonder, well, why are we doing all these different vaccines? What's the importance of that? Why would you just not come up with some standard approach? Well, different viruses tend to have different responses to different vaccine approaches, so it's probably a good thing that we're carrying forward with a whole bunch of different ones to see which one might be effective. The other thing that's questionable is, how long-term are the actual immunities that derive from these vaccines. There is some argument that live virus vaccines are better that way in terms of marshaling both innate as well as adaptive responses and good B cell and T cell responses. So these trials are really necessary to figure out what the best approach is. But it looks like the time frame of some of these vaccine approaches are much more rapid than have been talked about in terms of 12 to 18 months. They might be even much faster than that, which would be absolutely extraordinary if that's possible. And I think that's a relatively hopeful thing. I'm going to stop sharing my screen and just chat here for a little while. I think a couple of things that come to mind to me when I think about what's going on in the world. There really isn't a time, certainly in the last, I can't imagine 50 years, probably since World War II, where the entire scientific community has been focused on single problem. So any biomedical laboratory right now that can actually work on COVID-19 and SARS-CoV-2 is doing so. Every sort of laboratory, infectious disease laboratory in the world is trying to figure out ways to either generate therapeutics or a vaccine for this virus. And the therapeutic potential is also very high. And the reason I say that is that there are a number of antiviral drug candidates that are being tested right now. And there's a whole bunch of new ones that are being developed right now as well. And these are done based on very sophisticated, what's called combinatorial chemistry approaches, where they actually look at the structures of, for example, the spike protein of the virus and try to figure out small organic molecules that can bind in places that would prevent that virus from binding, of course, to the receptors on the cells. And those things are going on right now in huge ways all over the world. So my sense is that this level of scientific effort will yield good results. If only you could diminish the severity of the disease to a small extent by a therapeutic. In other words, the therapeutic doesn't have to be curative. If it can only reduce the severity of disease, we would lower the number of days of hospitalization. We would lower the number of patients that have to be mechanically ventilated. We'd have all kinds of good outcomes. And just that small change would actually change what's happening with the overwhelming of ERs and critical care units, right? So just a small benefit from therapeutics will actually be very, very beneficial in terms of the overall impact on health services. The other thing I'll say about COVID-19 is that there is no question that outside of age, the biggest risk factors, comorbidities, have to do with a lot of comorbidities that folks in the US live with, hypertension, obesity, diabetes, cardiovascular disease in general, lung dysfunction, these kinds of things that unfortunately the US population has a relatively high degree of a lot of this. And these are things that the SARS-CoV-2 virus seems to be particularly serious for individuals with those kinds of comorbidities. And as it happens, I think it's really highlighted some of the health disparities that exist in the United States now, particularly amongst minoritized communities, which have had historically much less access to healthcare, as well as access to healthy living conditions. So this is something that is highlighting disparities, both in terms of general health and welfare, as well as access to health, which is something that really has to be addressed over time. And I would echo Scout's admonition of thanks towards our healthcare professionals who are on the front lines working in hospitals across the country to deal with what has been a tremendously taxing, difficult, horrific pandemic for them in terms of trying to deal. And remember, the critical care folks are sort of evolving their knowledge to deal with this virus as they go along. And one of the things that's been clear is that there's a multi-system aspect of COVID-19. It's not just affecting the respiratory tract, it's affecting the kidneys, where it's clear that the virus can also infect cells. It's infecting the digestive tract, where it's clear that the virus can infect cells. The heart, where again, the virus can affect and have impacts on. The immune system is also impacting these because the immune system can over-respond and actually generate problems like clotting dysregulation and all of that. And so the multi-system aspects of the virus have proven to be very, very challenging. So now patients in critical care settings are going to have ventilators as well as dialysis machines, and it's really, really challenging. I'll end with the fact that when I was working at the State Department on pandemic influenza preparedness, we used to go to workshops on that H5N1. And I remember talking in a break once with Tony Fauci, who was there, of course, as a representative of National Institute of Allergy and Infectious Disease. And this was 15 years ago. And he was saying that to a group of us that he was very concerned about pandemic influenza because he said that we didn't have enough public health surveillance. We didn't have enough critical care beds in the United States. We didn't have enough good infection control in hospitals, in other words, isolation rooms and PPE, and that we didn't have enough ventilators. And so that's all proven to be true, of course. And so these problems in terms of our healthcare capacity and infrastructure have been known about for years and years. We just have not acted on them. And now it's showing this COVID-19 is actually highlighting a lot of these problems. So I'll take some questions now.
Thank you so much for your presentation, Professor Henson.
Yeah.
So we will now begin the question and answer session. And as a reminder to our viewers, you can comment any questions that you have in the chat box next to this live video. Depending on time, we may not get to all questions. So our first question is, can you comment on the possibility of this and other viruses of undergoing mutations that render them ineffective with binding onto human cells? And this individual was also wondering if you could clarify the immunological and biochemical basis of herd immunity.
Yeah, sure. Well, first, we'll do the herd immunity thing. Herd immunity just refers to the fact if you have a population of individuals, if enough of them have immunity to a pathogen, obviously it doesn't matter. So let's say we have 100 individuals and 60 to 70 of them have immunity to the pathogen. Well, the other 30 or 40 of them that don't have immunity to the pathogen, they probably are protected by the fact that the 60 or 70 do, right? Because the virus will not be spreading amongst individuals who have immunity to it. So herd immunity is simply this idea that once you get to a certain percentage of the population that's immune, the rest of the folks aren't as worried. And this is something that happens in the United States with vaccinations, right? There's enough folks that get measles, mumps, and rubella vaccines, enough kids who get it, that those kids who don't want to get it or their parents refuse to get it to them, they're protected by that. So herd immunity is something we would love to get to with COVID-19, but we're, as far as we can tell, we're far away from that, because even these tests that are done with antibody tests have suggested that maybe there's 20%, 15 to 20% in very hard hit areas. So we're far, as far as we can tell now, we're far away from herd immunity. In terms of the mutation rate of the virus, coronaviruses in general tend to be less subject to mutation over time than, let's say, influenza viruses. Influenza viruses mutate a lot, and that's why we have to have new vaccines every year. The virus is changing. So far, in looking at the sequences of this particular coronavirus, which is a sequence that looks like it came right from a bat, it's a native sequence. It's not a bioengineered virus. It's a virus that is a reservoir in bats. Might have an intermediary in other animals. Pangolins have been speculated on, but it isn't mutating a lot, which is good. And it turns out that the spike protein is pretty similar to the spike protein on SARS, the COVID-1 SARS. And it's a region that, in terms of its binding to the receptor, is relatively difficult to mutate in a way that wouldn't render it actually less virulent. It's probably as likely, remember, when viruses mutate, they're just as likely to become less virulent, in fact, more so in many respects, than more virulent, right? So if the virus mutates, it could mutate in a way in which it becomes much less virulent.
Thank you. So the next question is, this individual would like to know what is the cellular response difference between someone who recovers and someone that does not recover from the virus.
Versus recovers. So in terms of recovery, one would argue that the person who mounted a good, successful immune response would mean that they would have, of course, memory cells that they were generating that actually hang around. And in many cases, these memory cells can hang around for decades. So the B cells that had made antibodies and the T cells that had done their regulation and their killing of viral-infected cells, a subset of those would actually hang around. And folks have looked for these in SARS patients actually and found some examples of both B and T memory cells from SARS patients from 2003. And in some cases, the antibodies themselves are persistent, interestingly enough. In particular cases, there's been good validated research that's looked at antibody responses in patients who are survived certain influenza pandemics that had antibodies present even 50, 60, 70 years after initial insult. So immunological memory is quite powerful, and that would be the representative of a recovered patient. Individuals who don't recover, I assume they eventually are going to be killed by the virus, because I don't know another way that would happen. So those individuals are often individuals who don't mount necessarily the correct immune response. They might have comorbidities that make them much more fragile in terms of medically, in terms of, for example, lung function or cardiovascular health, or strictly age. If you look at viruses, respiratory viruses affecting, particularly in animal studies, animals of different ages, you'll get much more pathology in the older animals than you get in younger animals. It's just that way it is, the way it is.
Thank you. So the next question is, have you heard anything about treating COVID-19 with ivermectin?
Ivermectin, yeah, which is this drug, which is a drug used against parasitic infections all over the world. And I've heard, I haven't seen any, again, published data on that, but it's another one of these drugs that seems that people are trying everything out of the medicine cabinet to see if it works on COVID-19. You may have heard there's a clinical trial now of the active ingredient in the anti-heartburn drug Pepcid, because it's apparently a drug that has some efficacy. I remember we were talking about that protease that's involved in the entry of the virus into the cell, that apparently has some inhibitory response to that. So I get the sense that there's going to be a lot of folks trying a lot of different kinds of drugs. I don't know very much about ivermectin itself. The chloroquine and chloroquine, as far as the trials of that has happened, it seems like those drugs at least don't have what is obvious efficacy. In terms of their mechanisms, they're supposed to be drugs that interfere with the virus's ability to escape the endosome, the structure that the cell uses to bring the virus in once it binds to the receptor. So, but that doesn't seem to be working, at least in the trials that I've seen. So, remdesivir, there was a lot of hope for that because it's a nucleotide analog and it interferes with viral replication. Particularly, it's done so in MERS. In the MERS virus, it's been successfully looked at. And the first clinical trial was accidentally published by WHO, which didn't show any benefit, but there's other trials going on. So it's difficult to say. Now, the other thing that's going on, a lot of people are trying these drugs on very severely ill patients, and it's possible that it's too late really to treat them. So we'll see.
The next question is, what does the latest information on high blood pressure medications such as ACE inhibitors and ARBs tell us? Do they help diminish the severity of COVID-19 or exacerbate the disease?
Yeah, no, that's a great question. There's been a lot of speculation about that, of course. Many patients take ACE inhibitors as a way to manage blood pressure or blood pressure medications that are affecting the whole ACE system. And there's been some arguments that, of course, if you're inhibiting ACE, you probably are leading to the increase in expression of ACE on the surface of cells, which would at least arguably lead to a more vulnerable situation in terms of a virus actually binding to cells if you have more receptors there. But there really isn't a lot of evidence for that. In fact, there's some evidence in animals that there might be a protective function for ACE in terms of respiratory viruses. So there is a lot of back and forth in sort of the scientific community about whether ACE inhibitors are actually going to be protective or make patients more vulnerable. Right now, all the major medical societies and cardiology associations have come out saying that patients who are taking those drugs should continue to take them. There's no scientific evidence that tells those folks they actually to stop taking those drugs. And I would think that right now, as clinical, case studies are being done and they're releasing some of this clinical information, that they're going to have to start looking at, these drug-based comorbidities as well. So, for example, there's been some major studies that have been released the last week or so with 5,000 patients from New York, and they were looking at hypertension as a comorbidity, which is a very high one, as it turns out. And they're starting to look at drug, possibilities of particular drugs leading to severity of the disease. So I think we'll have to wait a little while to figure that out.
Great, thank you. So this next question is, where does the cytokine storm occur in the natural immune response?
Yeah, good question. So it turns out that, cytokines, of course, are just a general name for a lot of chemical factors that were released by cells that are part of the immune response. And so, for example, when a virus first infects your cells, one of the first cytokines that come out of these proteins that I've mentioned, interferons, right? So they're released by cells to try to interfere with the virus's ability to infect other cells. At the same time, macrophages that are actually ingesting infected cells or being responsive to virus themselves are also releasing cytokines. Interferon 1 is a classic one. It's interleukin 1. Interleukins, you can imagine white blood cells are often referred to as leukocytes. So the chemical factors that go between them are called interleukins. Interleukin 1 is a classic, one of the earlier interleukins. It actually is one of the febrile interleukins. In other words, it leads to fever responses. Also a lot of different responses. And then a whole host of other cells, from macrophages, the dendritic cells, to T cells, are releasing a lot of these cytokine factors too. Unfortunately, some of these factors, they're doing good because they're leading to vascular permeability in the capillaries to release fluid, so that things like complement, that are factors that can help kill viral infected cells, get in, antibodies get in. But also fluid tends to lead to leakage, places like the lung, which can be problematic, of course. They also lead to cells leaving the blood, so infiltrating into the tissues, and that's good in some respects, because those cells often have to eat other cells or kill other cells, but it also leads to problems, for example, in the lungs. And then once you get all these cytokines in your body at once, it can lead to whole body responses like hemorrhagic shock, where you have loss of fluid in the blood suddenly, or you might have clotting dysregulation, where clotting occurs now in low blood pressure conditions. And all these things seem to be happening, unfortunately, in some COVID-19 patients. So ideally, in the normal immune response, there isn't a cytokine storm. That it's usually cytokines are highly regulated, but it seems to be when you're in these acute radical infection situations like SARS-CoV-2 causes, these cytokine release syndromes actually develop. It's another focus of treatment, as it turns out. As many of you are aware, there's a lot of biologic pharmaceuticals, so often monoclonal antibodies, as it turns out, that are used to try to tamp down the immune response. So if you see these advertisements on TV for Humira or things like that, these are actually monoclonal antibodies that are trying to interfere with tumor necrosis factor alpha, which is another classic inflammatory cytokine. And so there's actually several clinical trials going on with COVID-19 now, which using antibodies to try to knock down cytokines, particularly a cytokine called interleukin 6, which is a very pro-inflammatory cytokine, and there's a specific antibody that can do that. It's used actually to treat autoimmune disorders. And so a lot of clinical trials are now focusing on trying to tamp down the immune response to try to help COVID-19 patients.
Thank you.
I should mention that H1N1, the 1918 influenza outbreak, it's thought that cytokine storm responses to that particular virus was probably as detrimental as any. And in fact, in that particular outbreak, a lot of young people died. In fact, people 60 and above did relatively well. People who are young, healthy, in their 20s and 30s and 40s were dying of that particular illness. Children to some degree as well. And possibly it was because it was the immune system over response that was doing more harm than good. So.
Thank you. So this next question is, for those people who have compromised immune systems, what are they lacking when it comes to the immunity cells? And does it vary according to what kind of autoimmune disease you have?
It does. And so there's a lot of different autoimmune disorders, of course. Some people might have problems with, for example, production of particular antibodies by B cells. Some folks can have issues with how many, for example, T helper cells they might have. Obviously, folks who have very severe combined immunodeficiencies are folks that who would have to be treated early in their lives to try to correct that, or else they wouldn't be able to live very long. And in fact, patients who have those kind of severe combined immunodeficiencies are often treated early in life with bone marrow transplants in an effort to try to repopulate all their immune cells with cells that do function properly. But yeah, there are autoimmune disorders that involve a lot of different conditions that can immunocompromise folks. The other thing that's happening in the US population, there's a lot of individuals who take drugs that immunocompromise them, right? That put them in a situation where they cannot fight off infections as well as they might ordinarily. And some of these are these biologics that, for example, are doing things to try to combat rheumatoid arthritis or lupus or these autoimmune disorders. So those individuals actually are tamping down their immune response with drugs, and that actually makes them more susceptible to pathogens than they ordinarily would be.
Thank you. So the next question is, what is causing the transfer between species? So recently with 3 tigers, 2 cats, and now a dog.
Yeah. It turns out that this particular virus is pretty broad in terms of its species specificity. So it can, in fact, in coronaviruses in general, been known to infect a number of different animals. As it happens, the species broadness of this particular virus, it can infect cats. Cats seem to be particularly susceptible, although largely, at least the things that I've seen in the veterinary work I've looked at, that cats tend to have mostly milder symptoms. It's not necessarily a high mortality virus for cats. And in that it's a virus that seems to have fairly broad species distribution, which may explain why, of course, humans got it from the bat. It's not clear how that actual transmission took place. I think there's a lot of speculation about humans' interactions with animals in the Wuhan market or maybe outside of Wuhan. There's been speculation about the virus escaping from a coronavirus laboratory in Wuhan itself. I will say that the probability of that is probably very low, although interesting historically, there have been viruses that have been first recognized after escaping from laboratory settings. Marburg is a classic, named after Marburg, Germany, a filovirus very similar to Ebola, which escaped from a laboratory that was working on African green monkey kidney cells. It was a filovirus. It's actually highly fatal in the 1960s. And of course, smallpox, which was a virus that was, of course, eradicated And that's one thing I think to keep in mind here. Smallpox, I'll just mention the last smallpox death was caused from a laboratory accident where smallpox came out of a laboratory. But I should mention that, this virus will not be eradicated in the same way that smallpox or polio could be if it's ever reached to that point, because smallpox and polio are viruses that only circulate in humans, whereas this virus has animal reservoirs, of course, and so it won't be eradicated per se. It could mutate in a way that makes it much less virulent, in which case we probably would just sort of forget about it, but it's not going to be, there's not going to be an end, or it's not going to just disappear like, let's say, an eradicated virus would.
Right. So this audience member wants to know, what do you believe are the top 2 or 3 things individuals can do to strengthen their immune systems generally?
Yeah, that's a great question. If we knew that, I'd probably make a lot of money if I knew exactly what it was that people needed to do. Yeah, so certainly, good diet, exercise is known to be one of these things. As they often say, exercise is the only magic bullet left. It is something that is known to boost the immune response to a certain degree. Elite athletes that exercise too much can actually lower their immunity, but in general, most of us, exercise is something that boosts immunity. You can also, of course, make sure you're getting all your vitamins and a good diet and all that. For a while there was some discussion about zinc as a possible way to lower infectivity of certain viruses, zinc supplements, but I think that the jury is still out on that. But yeah, it's funny, early on in January, I had an email interaction with an infectious disease physician who actually had worked on coronaviruses. This is way back in January when Wuhan was just starting to be an outbreak. And he, at that time, said that he laid out a series of things that he thought everybody should do. And that was everything that everyone's doing now, finally, which is wear masks, wear gloves, don't touch surfaces, all these things that we're doing now. He was arguing we should be doing, in January, which is interesting because he had worked in coronavirus, so he knew how actually transmissible those viruses could be, and he was absolutely right. So we finally gotten to the point where we're, done what has to be done, but it's taken a while.
Great. Now, Professor Henson, I know we've technically reached the end of our time.
Yes.
We do have 3 or 4 more questions in the chat. I want to be respectful of your time. Do you have the time to answer these questions?
Sure.
Or okay,
Why
so I'm
not?
going to end it after the ones that I see at this moment now.
Sure.
So our next question is, what causes someone to be asymptomatic but still a carrier?
Yeah, no, it's a good question. I don't think we really know the answer to that. There's been some suggestion that asymptomatic individuals aren't getting a very mild level of viral load and might not even be generating a significant immune response, but at the same time shedding virus. But I just don't think we know enough about what's happening with those individuals. I don't think we even know what percentage of individuals are asymptomatic. There's some antibody-based testing that's been done in Germany in particular that suggests it might be relatively high, but it's very difficult to say because the specificity of some of those tests, as we all know, is not great, at least some of them, so we'll have to see.
Thank you. So this next question, you mentioned this a little bit earlier, but I'm wondering if you can expand on it. The question is, can you hypothesize the potential reasons for why it appears that children are less affected by this virus compared to adults, unlike other viruses?
Yeah, that's another real big mystery. I was just reading a commentary paper on this subject, where one of the hypotheses is that, well, perhaps the immune system is not as well developed in children, and therefore these problems that come post-infection with immune overreaction aren't taking place as much. Maybe there's a slight difference in the expression of some of these receptor proteins in the respiratory epithelium of children versus adults. There's a lot of hypotheses about it, but I'm not aware of any specific ideas. There also is, of course, this gender specificity to the seriousness of the illness, and that's also been a lot of work done on that. And males are disproportionately affected, of course, and some of those arguments revolve around males having worse health-related behaviors than females. Others, there's a well-established studies over the years that have shown that females have better, in general, immune responses to viruses than males do. And part of that might be an evolutionary thing in terms of evolution driving the preservation of gender that's carrying forth the delivery of the future generations. Of course, females pay a price for that because they have a disproportionate representation of autoimmune diseases as well, which is kind of interesting. But anyway.
Yeah, that is interesting. Thank you. So this next question is, how reliable are the antibody tests in predicting acquired immunity?
Yeah, it's a tough one too. You probably heard on the news, the FDA gave emergency, sort of approval to a lot of these tests, and then a group at UCSF decided to test them, and they tested like 12 or 13 of them and only found a subset of them that were reasonably reliable. But I mean, the encouraging thing is that they found some that were reliable, and this reliability we're talking about in the 90th percentiles or something like that. And my sense at this point is because we have so little knowledge about general, population-based testing, that if we have an antibody test that's, 90% effective or specific, I think that is certainly worth pursuing because right now we have zero knowledge about a lot of what's going on in population-based immunity to this virus. So having even a less than ideal antibody test, for me at least, would make sense. I think that's the only way we're going to get at that question, because the RT-PCR, diagnostic test for active virus is a complicated test, requires machinery, requires trained technicians. It's a difficult thing to do millions and millions of tests. The antibody tests are strips of paper, they're lateral flow assays that can be done at home. So they would be the way to scale up testing. And let's face it, the two choices we have are to do, as we go into the fall, for example, where the virus is still going to be here, it's going to wax and wane. The two choices we have are to test and trace to the maximum we can do in order to keep the virus in check, or continue to strangle the economy of the United States and the world with this situation of social distancing we're in right now. I think with that choice, anybody would say, well, we're going to have to test and trace. That's all we can do. I mean, that's what has to be done. So,
right. So this will be our last question. This audience member commented, there are trials using mesenchymal stem cells as a potential treatment for COVID-19. What is the mode of action for this treatment?
Yeah, mesenchymal stem cells. I'm not certain. I did see some mention of these stem cell trials, but I did not look at the mechanism for how those are working. I'm not even clear what would be going on in those particular situations, but yeah, I don't know.
Thank you.
Okay.
So this will conclude tonight's program. Thank you so much, Professor Henson, for talking with us tonight.
Sure.
And thank you to our audience members for tuning in. Have a great night, everyone.
All right, good night.